In the early years of the 20th century, in a house in Notting Hill where books lined the walls and conversation flowed like water, finding its natural channels, a girl was born who would one day hold the secret of life itself in her hands. Rosalind Elsie Franklin arrived on July 25, 1920, the second of 5 children in a family that valued education the way others valued inherited wealth, as the foundation upon which everything else could be built. The Franklin family was Jewish, though not strictly observant. They were prosperous, though not ostentatiously wealthy. They believed in service, in contribution, in using privilege to improve the world, rather than merely to accumulate comfort. Ellis Franklin, Rosalind's father, was a merchant banker who also taught at the Working Men's College, bringing education to those whom society had denied it. Muriel Franklin, her mother, managed the household with quiet efficiency and devoted herself to causes that aided those in need. The children were expected to think, to question, to defend their positions with evidence and logic. Young Rosalind moved through this environment, absorbing its values. She was not the easiest child. She was stubborn, precise, intolerant of imprecision in herself or others. When she argued with facts. When she was wrong, she required proof before conceding. These traits, which in a boy might have been celebrated as signs of intellectual promise, were sometimes viewed with concern in a girl. But the Franklin household, for all the limitations of its era, recognized intelligence when it appeared. Rosalind was given the same educational opportunities as her brothers. At St. Paul's Girls' School, she discovered science, not as a subject to be memorized, but as a way of seeing, a method for asking questions and demanding that the universe answer with evidence rather than assumption. Chemistry and physics became her languages. The laboratory became her sanctuary. While other girls her age navigated the social complexities of adolescence, Rosalind navigated equations, finding in their precision a clarity that human relationships often lacked. She was not without friends. She was not without warmth. But she expressed these qualities differently than expected, and those who did not look carefully sometimes missed them entirely. She loved hiking in the mountains. She traveled when she could, exploring landscapes with the same curiosity she brought to molecular structures. She maintained lifelong friendships with those who understood her, who recognized that her directness was not coldness, but honesty, that her high standards were applied most rigorously to herself. In 1938, she entered Newnham College, Cambridge, to study chemistry. The timing could not have been more fraught. Europe was sliding toward war. Hitler had annexed Austria. The Munich Agreement offered false hope of peace. And at Cambridge, women occupied an uncomfortable position, allowed to attend lectures and take examinations, but not permitted to receive actual degrees. The university would not grant degrees to women until 1948, years after Rosalind had completed her studies and distinguished herself in research. She would eventually receive her degree retroactively, a correction that came too late to change the experience of having earned recognition that was officially denied. The war arrived during her second year. The Blitz began in 1940, raining bombs on London, destroying buildings, killing thousands. The Franklin family evacuated, scattered to safer locations. Rosalind remained at Cambridge when she could, studying with an intensity that might have seemed strange if one did not understand that for her, the work was not escape from reality, but engagement with it. Understanding the physical world was not separate from understanding the human world. Both required careful observation, both required evidence, both required the humility to admit what one did not yet know. She graduated in 1941 with second-class honors, a result that disappointed her, that she felt did not reflect her abilities. But the degree classification would prove irrelevant to her actual career. What mattered was what she learned, what she could do, what problems she could solve. After graduation, she worked briefly at the British Coal Utilisation Research Association, studying the physical structure of coal and carbon. It was not glamorous work. It was not the kind of research that appeared in newspaper headlines. But it was meticulous, it was useful, and it taught her techniques that would later prove essential. She learned about crystallography, the science of understanding molecular structure by analyzing how X-rays scatter when passed through crystalline materials. The patterns they create, the angles and intensities of diffraction, can be read like a language by those trained to interpret them. Each pattern speaks of the arrangement of atoms, of the architecture of matter too small to see directly. The coal research earned her a doctorate from Cambridge in 1945. Her thesis examined the porosity of coal at the molecular level, how tiny holes in the carbon structure affected its properties, its usefulness as fuel, its behavior under different conditions. The work was solid, was respected, established her reputation as a careful experimentalist. But it was only beginning. The techniques she had mastered would find their true application elsewhere, in a molecule far more significant than carbon, in a question far more profound than fuel efficiency. The war ended. The world began the long process of reconstruction, and Rosalind Franklin, newly minted Doctor of Philosophy, looked for the next problem worthy of her attention. She found it in Paris, in a laboratory that would welcome her in ways England had not, where her skills would be valued and her gender would be somewhat less of an obstacle. She was 26 years old. She had survived the war. She had earned credentials that should have opened every door. And she was about to enter the happiest years of her professional life in a city still recovering from occupation, where the collaboration she found would sustain her through the more difficult years to come. The daughter of light and shadow, already shaped by precision and patience, already trained to see what others could not see, already becoming the scientist who would one day aim X-rays at the molecule of life and capture its shape in patterns of darkness and light. The chapter closes. London recedes. Paris waits. and Rosalind Franklin steps forward into the next phase of a journey whose destination she could not yet imagine: Paris. In the years after liberation was a city learning to breathe again. The occupation had ended, but its shadows lingered in empty chairs at family tables, in buildings still bearing bullet scars, in the collective memory of collaboration and resistance that would take generations to process. Yet there was also renewal, an insistence on life continuing, on culture reviving, on science resuming the questions that war had interrupted. Into this atmosphere came Rosalind Franklin in 1947, taking a position at the Laboratoire Central des Services Chimiques. She was 27 years old. She spoke French fluently, having learned it as a child during family holidays, and polished it through continuing study. She was ready for something new, something that would challenge her, something that would allow her to grow beyond what English laboratories had offered. What she found exceeded her hopes. The French scientific community welcomed her in ways she had not experienced in England. Here, her gender was obstacle less formidable. Here, her precision and her directness were valued rather than viewed with suspicion. Here, she found colleagues who became friends, who invited her into their homes and their conversations, who treated her as what she was, an exceptionally talented scientist whose contributions mattered. She worked on the structure of carbon, continuing the research she had begun during the war, but with new techniques and new questions. X-ray crystallography became her primary tool. The method was elegant in concept. Pass X-rays through a material, observe how the rays scatter as they encounter atoms, and from the patterns of scattering, deduce the arrangement of atoms within the material. In practice, the method was extraordinarily demanding. The mathematics were complex. The experimental conditions had to be precisely controlled. The interpretation of results required both technical skill and intuitive understanding of how molecular structures produce particular diffraction patterns. Franklin mastered all of it. She refined techniques for preparing samples, for positioning them in the X-ray beam, for capturing the diffraction patterns on photographic plates. She developed expertise in interpreting those patterns, in translating the abstract geometry of scattered X-rays into concrete knowledge of molecular structure. The coal and carbon work she published during these years established her international reputation as a crystallographer of exceptional ability. But the work was more than technical accomplishment. It was a kind of seeing, a way of perceiving the hidden architecture of matter that most humans would never glimpse. The molecules she studied were far too small to observe directly. No microscope could resolve individual atoms in a carbon structure. But X-rays could illuminate what eyes could not see, could reveal the patterns that atoms create when they arrange themselves in crystalline order. To master crystallography was to develop a sixth sense, a capacity to perceive the invisible through its effects on light. The years in Paris were happy years. Franklin hiked in the Alps during holidays. She maintained correspondence with family and friends in England. She attended conferences and met scientists from across Europe who shared her passion for understanding the physical world. She was productive, publishing papers that advanced understanding of carbon structure and demonstrated her technical mastery. She was valued, respected, included. The laboratory became a kind of home. The colleagues became a kind of family. The work became a kind of prayer, patient, meticulous, devoted to truth that could be verified rather than merely believed. She was building something during those Paris years, not just publications and reputation, but a sense of herself as scientist, as someone capable of seeing what others could not see and explaining what she had seen so that others could understand. Jacques Mering, her supervisor and mentor, recognized her abilities and encouraged her development. The relationship was collegial, productive, supportive in ways that later English experiences would not replicate. Mering taught her not just techniques, but confidence. Confidence that her observations were valid, that her interpretations were sound, that she had every right to contribute to scientific conversations as an equal. She could have stayed in Paris. She was happy there. The position suited her, the colleagues valued her, the city delighted her. But in 1950, she learned of a fellowship available at King's College London, an opportunity to apply her crystallographic skills to a new problem, the structure of biological molecules, particularly DNA. The decision to return to England was not simple. She knew what English scientific culture was like, knew that women faced obstacles in London that they did not face in Paris. But the problem was compelling. DNA, deoxyribonucleic acid, was known to carry genetic information, but its structure was unknown. Understanding that structure might reveal how inheritance worked at the molecular level, how the information in genes was encoded and transmitted. It was a problem worthy of her abilities. She accepted the fellowship. She prepared to leave Paris, to leave the colleagues who had become friends, to leave the city that had welcomed her. She did not know what awaited her in London. The tensions, the miscommunications, the photograph that would change everything, and the recognition that would never come. The chapter closes in the Paris laboratory, equipment humming softly, X-ray photographs drying in the dark room, Franklin gathering her notes and her expertise for the journey home. The carbon work had taught her patience. It had taught her precision. It had taught her to see the invisible through its effects on light. Now she would turn those skills toward something new, something that would reveal not just the structure of a molecule, but the architecture of life itself. The cathedral of carbon had prepared her. The helix waited. King's College. London rises along the Thames, its neoclassical facade speaking of tradition and institutional weight. In 1951, when Rosalind Franklin arrived to take up her fellowship, the college was still recovering from war damage, still negotiating the awkward transition from wartime disruption to peacetime routine. The biophysics unit, where she would work, occupied basement laboratories, appropriate perhaps for research that delved into the hidden structures of existence, but hardly welcoming. She came with specific expectations. John Randall, the head of the unit, had written to her describing a project she would lead, using X-ray crystallography to study the structure of DNA. She understood this to mean she would work independently, applying her expertise to a problem that had not yet been solved. She arrived ready to establish her laboratory, to train any assistance she might need, to pursue the research with the precision and rigor that had characterized her Paris years. What she found was more complicated. Maurice Wilkins, another researcher in the unit, had already been working on DNA crystallography. He had assumed that Franklin would join his project as a junior colleague or collaborator. The miscommunication, whether Randall's failure to clarify roles or simple institutional dysfunction, created friction that would persist throughout Franklin's time at King's. The tension was not merely administrative. Wilkins and Franklin were profoundly different in temperament. He was diffident, indirect, inclined to suggest rather than state. She was direct, precise, impatient with ambiguity. He found her cold and difficult. She found him presumptuous and unclear. They were both wrong, and they were both partly right, and they were both trapped in a dynamic that neither fully understood and neither could escape. But the work continued despite the tension. Franklin set up her X-ray equipment. She refined techniques for preparing DNA samples, thin fibers that could be mounted in the X-ray beam, kept at precise humidity to maintain their crystalline structure. The conditions mattered enormously. DNA existed in different forms depending on moisture content, and the patterns produced by different forms were different. Understanding which form you were studying and maintaining consistent conditions throughout long exposures required exacting attention to detail. Raymond Gosling, a graduate student who had been working with Wilkins, was assigned to work with Franklin. The reassignment added to the awkwardness. Gosling was now caught between two researchers who could barely communicate with each other. But Gosling himself proved to be a capable and congenial assistant. Together, he and Franklin pursued the crystallographic work with increasing sophistication. The DNA they studied came from calf thymus glands, purified and drawn into thin fibers that could be mounted in the X-ray apparatus. The fibers were remarkably uniform, testament to DNA's regular structure, and produced surprisingly clear diffraction patterns. This clarity was itself significant. It suggested that DNA had a highly ordered, repetitive structure, not the irregular arrangement that some scientists had assumed. Franklin distinguished between two forms of DNA, which she labeled A and B. The A form existed at lower humidity and produced a different diffraction pattern than the B form, which existed at higher humidity. Both forms were helical. She determined this from the patterns, but the details of their structures differed. She focused initially on the A form, which produced sharper patterns that she believed would be easier to analyze mathematically. The work was slow. It had to be slow. Each X-ray photograph required hours of exposure. Each photograph then required painstaking analysis, measuring the positions and intensities of diffraction spots, calculating what molecular structure could produce such a pattern. Franklin was building her case carefully, accumulating evidence, avoiding the speculation that might lead to error. Meanwhile, in Cambridge, James Watson and Francis Crick were pursuing DNA structure with different methods. They built physical models, metal and wire constructions representing atoms and bonds, trying to find arrangements that were chemically plausible and consistent with available data. Their approach was faster, more intuitive, more willing to leap ahead of evidence. It was also more collaborative, drawing on information from multiple sources, including information that had not been freely shared. Franklin did not know the Cambridge researchers well. She did not know that her work would become central to their efforts. She continued her research at King's, isolated by the tensions with Wilkins, focused on the precise and demanding crystallography that was her particular gift. The chapter pauses here, in the basement laboratory, the X-ray tube humming, the photographic plates accumulating evidence that would prove momentous. Franklin at her calculations, Gosling at his instruments, the DNA fibers mounted and waiting for the light that would reveal their secrets. The photograph that would change everything had not yet been taken, but the conditions for its creation were being established, the techniques refined, the samples prepared, the expertise accumulated. The secret was waiting to be revealed to eyes trained to see it. In May of 1952, Rosalind Franklin and Raymond Gosling captured an image that would become one of the most famous photographs in the history of science. Photograph 51, so numbered in their laboratory records, showed an X-ray diffraction pattern of the B form of DNA. The image was remarkably clear, a pattern of dark spots arranged in an X shape against a lighter background with a distinctive cross at the center and layer lines indicating regular spacing along the molecule's length. To untrained eyes, the photograph appears abstract, almost meaningless, spots and shadows on a grayscale field. But to crystallographers, it spoke volumes. The X-shaped pattern was characteristic of helical structures. The spacing of the spots indicated the dimensions of the helix, its diameter, the distance between turns, the number of chains it might contain. The photograph was, in effect, a shadow cast by DNA's molecular architecture, and those who could read such shadows could determine the structure that cast them. Franklin could read it. She recorded in her laboratory notebook that the B form was helical, with a diameter of about 20 angstroms and a spacing of 34 angstroms between complete turns of the helix. She noted that the phosphate groups, part of DNA's backbone, were on the outside of the structure, exposed to the surrounding water. These observations, derived from careful analysis of photograph 51 and related images, were substantial steps toward understanding DNA's structure. But she was cautious. She did not publish immediately. She continued accumulating evidence, refining her measurements, building the mathematical case that would support definitive conclusions. The A form still demanded attention. Its diffraction pattern was different, suggesting a different arrangement, and understanding how the two forms related required careful analysis. The caution was characteristic. Franklin did not speculate beyond her evidence. She did not claim certainty when uncertainty remained. This rigor, which in other contexts might be celebrated as scientific virtue, would prove costly in the race to determine DNA's structure. Others were less cautious, more willing to leap ahead, more eager to claim priority. The photograph sat in her laboratory files. She shared it with Gosling, who had helped take it. She discussed it with colleagues in the biophysics unit. She presented her findings at seminars, displaying her X-ray images and explaining what they revealed. But she did not broadcast her work widely. She was building toward publication, following the normal processes of scientific communication, unaware that time was running out. In January 1953, Maurice Wilkins showed photograph 51 to James Watson during a visit to King's College. The showing was not authorized by Franklin. The photograph was her work, her data, produced by her techniques and her analysis. Wilkins later claimed he did not realize he was doing anything improper. Franklin was leaving King's, and he may have thought her work was now common property of the unit. But whatever his understanding, the effect was the same. Watson saw the photograph and understood immediately what it meant. Watson was not a crystallographer. He could not have produced such an image himself, could not have refined the techniques required to capture it. But he knew enough to recognize what the X-shaped pattern indicated. He saw the helix. He saw the dimensions. He saw, in that single photograph, the key to the structure that he and Crick had been pursuing by other means. He returned to Cambridge. Within weeks, Watson and Crick had built their famous model, 2 helical chains wound around a common axis, with the phosphate backbones on the outside and the bases, adenine, thymine, guanine, cytosine, pairing on the inside according to specific rules. Adenine paired with thymine, guanine paired with cytosine. The complementary pairing explained how DNA could replicate. The 2 strands could separate, and each could serve as a template for building a new complementary strand. The model was elegant. It was correct. It was also built substantially on Franklin's data, not just photograph 51, but also the density measurements and structural parameters that she had determined and that had been shared, again without her knowledge, through a Medical Research Council report. Franklin learned of the Cambridge model in March 1953. She was invited to see it, traveled to Cambridge, examined it with her crystallographer's eye. By her own account, she recognized its plausibility. the model was consistent with her data. The structure it proposed could produce the diffraction patterns she had observed. She did not protest. She did not claim that her data had been used without permission. She continued her own work, preparing papers that would present her crystallographic evidence for DNA structure. Her papers were published alongside Watson and Crick's famous paper in Nature in April 1953. But the arrangement was clear. Watson and Crick proposed the structure. Franklin and Wilkins provided supporting evidence. The photograph that held the secret had revealed its secret. But the credit flowed elsewhere, to men who had seen what she had seen only because they had been shown her work without her consent. The double helix model proposed by Watson and Crick in 1953 became one of the most celebrated discoveries in the history of biology. The paper they published in Nature was brief, barely more than a page, but its implications were vast. The structure explained how genetic information could be encoded in the sequence of bases along the DNA strand. The complementary pairing of bases explained how that information could be copied when cells divided. The helical geometry explained how the long molecules could be compacted into the tiny space of a cell nucleus. But the discovery was not theirs alone. It emerged from data and insights contributed by multiple scientists, including Rosalind Franklin, whose photograph had provided the crucial evidence for helical structure. Understanding how credit was distributed and misdistributed requires examining how the work unfolded and how its history was later told. Watson and Crick were brilliant. This must be acknowledged. Their ability to synthesize information from multiple sources, to build models that integrated chemical constraints with physical data, to see the implications of complementary base pairing, these were genuine intellectual achievements. They were also skilled at the social dimensions of science, at cultivating relationships, at positioning themselves at the center of conversations about DNA structure. But they did not work in isolation. They drew on Erwin Chargaff's discovery that adenine and thymine and guanine and cytosine occurred in equal amounts in DNA, a clue to complementary pairing. They drew on Linus Pauling's work on helical structures in proteins and on the fear that Pauling might solve DNA's structure before they did. And they drew, crucially, on Franklin's crystallographic data that reached them through channels she had not sanctioned. The pathways of information transfer were multiple. Wilkins showed Watson photograph 51 in January 1953. Max Perutz, who supervised Crick, shared with him a Medical Research Council report containing Franklin's density measurements and structural parameters, information that helped constrain the model they were building. The sharing was not necessarily malicious. Perutz later said he did not realize the report was confidential. Wilkins may have thought Franklin's departure from King's made her work available to the unit. But intentions matter less than effects. The effect was that Franklin's data supported a model built by others who received credit she did not share. Franklin herself did not protest publicly. She continued her work, published her crystallographic evidence, moved to Birkbeck College to study virus structures. Whether she fully understood how her data had been used is unclear. Whether she would have protested if she had understood is equally unclear. She was not someone who sought conflict or controversy. She was someone who believed that scientific evidence should speak for itself, that credit would follow contribution, that the community of scientists would recognize good work and assign recognition fairly. She was wrong about that. Or rather, she was right in principle, but wrong about how principles operated in practice. Scientific communities are human communities, subject to the same biases and power dynamics that shape all human institutions. Women's contributions were systematically undervalued. Those who were well connected received more recognition than those who were isolated. Those who announced results confidently received more attention than those who presented evidence carefully. James Watson's memoir, The Double Helix, published in 1968, cemented a particular version of the discovery story. The book was vivid, irreverent, compelling, and deeply unfair to Franklin. Watson portrayed her as Rosie, a nickname she never used, describing her appearance with condescension and her manner with barely concealed contempt. He depicted her as an obstacle rather than a contributor, as someone whose data was valuable but whose judgment was unreliable. The memoir was controversial even at the time. Crick distanced himself from its portrayal of Franklin. Wilkins was unhappy with aspects of its narrative. Scientists who had known Franklin objected to its characterization. But the book became a bestseller, shaping public understanding of DNA's discovery for decades. The damage was done. Franklin was remembered, when she was remembered at all, as the difficult woman who almost got in the way of the brilliant men who solved the structure. Only gradually did the historical record begin to be corrected. Feminist scholars recovered Franklin's story. Biographers examined her notebooks and correspondence. Scientists insisted on accurate attribution. The process took decades and remains incomplete, but the truth is now available to those who seek it. Photograph 51 was Franklin's work. The data that constrained the model was Franklin's data. The discovery of DNA's structure was collaborative, and she was a central collaborator whose contribution was taken without acknowledgement. Step back now from the human story, the tensions and the credits and the recognitions denied, and contemplate what was actually discovered. The double helix is not merely a shape. It is the physical medium of inheritance, the molecule that carries the instructions for building every living thing, the chemical thread connecting you to your parents and your children, and every organism that has ever lived on Earth. DNA consists of two strands wound around each other in a spiral. Each strand is a chain of nucleotides, molecular units consisting of a sugar, a phosphate group, and a nitrogenous base. The sugars and phosphates form the backbone of each strand, running along the outside of the helix. The bases, adenine, thymine, guanine, and cytosine point inward, forming the rungs of the ladder, each rung consisting of a pair of complementary bases, adenine always pairing with thymine, guanine always pairing with cytosine. The complementary pairing is the key to everything. Because adenine pairs only with thymine and guanine only with cytosine, each strand of DNA contains all the information needed to reconstruct the other strand. When cells divide, the two strands separate, and each serves as a template for building a new complementary strand. The result is two complete double helices, each identical to the original. This is how genetic information is copied, how inheritance works at the molecular level, how the instructions for building an organism pass from parent to offspring. The instructions themselves are encoded in the sequence of bases along the strand. The sequence ATCG-ATCG carries different information than the sequence GCTAGCTA. The four bases serve as a four-letter alphabet, and their sequences spell out the instructions for building proteins, the molecular machines that carry out most of life's functions. The genetic code, the rules by which base sequences are translated into protein sequences, would be worked out in the decades following the structure's discovery. But the structure itself revealed how information could be stored in a chemical molecule, how it could be copied, how it could be inherited. The implications were vast. Understanding DNA's structure opened the door to molecular biology, the study of life at the level of molecules and their interactions. It enabled genetic engineering, the deliberate modification of organisms by altering their DNA. It made possible the biotechnology industry, the Human Genome Project, personalized medicine, forensic DNA analysis, and countless other developments that have transformed human life. Franklin glimpsed this structure in her X-ray photographs. She saw the helix in the patterns of diffracted light. She measured its dimensions, determined the positions of its phosphate groups, accumulated the evidence that would confirm its architecture. Her seeing was technical. She read diffraction patterns, not base sequences, but it was also profound. She perceived the shape of inheritance, the geometry of biological information, the molecular form that underlies all living things. The helix has become an icon. It appears in logos and sculptures, in museum exhibits and pharmaceutical advertisements. Its elegant spiral has come to symbolize biological science itself, the capacity to understand life at its most fundamental level. But icons can obscure as well as illuminate. The helix was not discovered by a single flash of insight. It emerged from decades of work by many scientists, from accumulated evidence and refined techniques, from collaboration and competition and the slow accumulation of knowledge that characterizes science at its best. Franklin's contribution to that emergence was essential. Without her photographs, without her data, the model could not have been built when it was built. The helix that represents modern biology bears her fingerprints, whether or not her name appears alongside it. In the spring of 1953, as Watson and Crick were celebrating their model and preparing their publication, Rosalind Franklin was preparing to leave King's College, London. She had accepted a position at Birkbeck College, also in London, where she would turn her crystallographic skills toward a new problem: the structure of viruses. The departure was not dramatic. There was no public confrontation, no formal accusation of stolen credit or professional mistreatment. Franklin simply moved on, leaving behind a laboratory and colleagues with whom she had never established comfortable working relationships, moving toward a new institution where she hoped to find better circumstances. The decision had been made before the DNA structure was announced. Franklin had grown tired of the tensions at King's, the isolation, the sense that her work was undervalued and her presence unwelcome. The fellowship that had brought her there was ending, and she had little desire to seek its extension. Birkbeck offered something different, a chance to build her own research group, to pursue questions that interested her, to work with colleagues who might prove more congenial than those at King's. John Desmond Bernal, the head of crystallography at Birkbeck, welcomed her. Bernal was a legendary figure in X-ray crystallography, politically radical, intellectually brilliant, known for supporting women scientists at a time when few men did. Under his sponsorship, Franklin established a research program studying the structure of tobacco mosaic virus, a pathogen that infected tobacco plants and that had become a model system for understanding viruses in general. The virus work was important. Tobacco mosaic virus consists of a protein coat surrounding a strand of RNA, genetic material similar to DNA, but single-stranded. Understanding how the protein subunits assembled to form the viral coat, how the RNA was positioned within that coat, how the virus infected cells and replicated, all these questions had implications for understanding viruses more generally, including viruses that caused human disease. Franklin approached the work with her characteristic precision. She refined techniques for preparing virus samples suitable for X-ray crystallography. She accumulated diffraction data, analyzed patterns, built understanding of viral architecture. She published papers that established her as a leading researcher in structural virology. She trained students and postdoctoral researchers, building a research group that would continue her work. The years at Birkbeck were productive. They were also, by many accounts, happier than the King's years. The tensions that had plagued her earlier position were absent. Bernal valued her contributions. Her colleagues respected her expertise. She found something closer to the collaborative environment she had experienced in Paris, something that allowed her abilities to flourish without constant friction. She traveled internationally. She visited laboratories in America, collaborated with researchers at Berkeley and Yale, presented her findings at conferences, established herself as an important figure in the emerging field of structural biology. The recognition she received for her virus work was genuine and appropriate. Recognition based on published evidence, on demonstrated expertise, on contributions that could be verified and replicated. But beneath the surface of professional success, something was wrong. She experienced abdominal pain that she attributed to work stress or digestive problems. She ignored symptoms, postponed medical attention, focused on research that seemed more urgent than physical complaints. The human body was not her area of expertise. She studied viruses, not herself. The chapter closes with Franklin at Birkbeck, surrounded by X-ray equipment and virus samples, her research group growing, her publications accumulating, her international reputation expanding. The shadow that would consume her had not yet been named, but it was already present, already growing, already marking the time she did not know was limited. Tobacco mosaic virus became her new cathedral. Under the electron microscope, it appeared as a hollow rod, a cylinder of protein surrounding a coiled strand of RNA. But the electron microscope showed only surface appearance. To understand how the virus was constructed at the molecular level, how protein subunits fitted together, how RNA was positioned within the structure, required X-ray crystallography, required the techniques Franklin had mastered during her years studying coal and DNA. She refined those techniques for viral work. Virus particles were larger and more complex than the molecules she had studied previously, presenting new challenges for sample preparation and data analysis. But the principles were the same. Pass X-rays through ordered arrays of particles, capture the diffraction patterns, analyze those patterns to determine structure. The tobacco mosaic virus proved particularly suitable for crystallographic study. Its protein subunits were arranged in a regular helix, creating the ordered structure that X-ray crystallography required. Franklin determined that 16 protein subunits made up each turn of the helix, an unusual number that reflected the geometric constraints of packing proteins around a central RNA strand. She mapped the positions of protein subunits, demonstrated that the RNA followed a helical path within the protein coat, contributed to understanding of how viruses assembled from their component parts. The work had practical implications. Understanding viral structure was prerequisite to understanding viral function, how viruses infected cells, how they replicated, how they caused disease. The tobacco mosaic virus was not itself a human pathogen, but the principles Franklin elucidated applied to viruses generally, including those that caused human illness. She expanded her research to other viruses. Poliovirus, which caused the feared paralytic disease, became a subject of study. Her research group examined plant viruses, animal viruses, bacterial viruses. The structural approach she championed was proving fruitful across the entire range of viral biology. In 1956, she was diagnosed with ovarian cancer. She was 36 years old. The cause is unknown. Ovarian cancer has multiple risk factors, and attribution to any single exposure is impossible. but she had worked with X-rays for years, in an era when radiation safety protocols were less rigorous than they would later become. Whether the exposure contributed to her illness cannot be determined with certainty. What is certain is that the diagnosis was devastating. Yet she continued working. Through surgery and chemotherapy, through the debilitating effects of treatment, she maintained her research program. She supervised students, attended conferences, published papers. She did not withdraw from scientific life. She pursued it with the same intensity she had always brought to her work, as if the research itself could hold back the disease that was consuming her. Her colleagues at Birkbeck were supportive. Bernal and others ensured that she had the resources and assistance she needed to continue. Her research group rallied around her, completing projects she could no longer manage alone, carrying forward work she had initiated. The community that had sometimes failed her earlier now supported her in her final years. She traveled to America even as her health declined. She presented research at scientific meetings, discussed virus structures with colleagues, maintained the international connections she had built. She refused to let illness define her, refused to become a patient rather than a scientist, refused to surrender the identity that work had given her. The light was fading. She knew it was fading. But she worked until she could work no longer, leaving behind research that others would complete, publications that established foundations for future discoveries, a scientific legacy that extended far beyond the DNA work that would eventually make her famous. On April 16, 1958, Rosalind Franklin died at the Royal Marsden Hospital in London. She was 37 years old. The ovarian cancer that had been diagnosed 2 years earlier had spread despite treatment. She had continued working until weeks before her death, maintaining the focus and precision that had characterized her entire career. 4 years later, in 1962, the Nobel Prize in Physiology or Medicine was awarded to James Watson, Francis Crick, and Maurice Wilkins for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. Rosalind Franklin was not mentioned. The Nobel Prize is not awarded posthumously. This policy meant that Franklin could not have been included in the 1962 prize, regardless of how her contribution was assessed. But the policy does not address whether she would have been included had she lived. The Nobel Prize is limited to 3 recipients per award, and the DNA structure had 4 principal contributors. Someone would have been excluded. The question of who that would have been remains speculative. Would the committee have recognized Franklin's essential contribution, her photograph that revealed the helix, her data that constrained the model? Or would she have been passed over in favor of the men who built the model and announced its implications? The history of Nobel Prizes suggests that women's contributions to scientific discoveries have been systematically undervalued, that women have been excluded from prizes for work they performed, that the awards reflect the biases of those who bestow them. What is not speculative is how the discovery was remembered and celebrated. Watson's memoir, The Double Helix, published in 1968, became the canonical account. Its portrayal of Franklin, rosy, difficult, obstructive, valuable only for her data, shaped public perception for decades. The book was enormously popular, reaching audiences far beyond the scientific community, establishing a narrative that elevated Watson and Crick while diminishing Franklin. Francis Crick later acknowledged Franklin's contribution more generously, noting that her X-ray photographs had been crucial to their work. But acknowledgments published in scientific papers or spoken at conferences do not have the cultural reach of best-selling memoirs. The popular understanding of DNA's discovery continued to feature Watson and Crick as heroes, with Franklin as footnote or obstacle. The correction has been slow. Anne Sayre's biography of Franklin, published in 1975, challenged Watson's portrayal and presented Franklin as the capable scientist she was. Brenda Maddox's biography, published in 2002, provided comprehensive documentation of Franklin's work and its appropriation. Feminist scholars analyzed how gender bias shaped both the original distribution of credit and its subsequent retelling. Scientists who had known Franklin and scientists who had simply studied the evidence insisted on accurate attribution. Today, Franklin is recognized. Her name appears alongside Watson, Crick, and Wilkins in many accounts of DNA's discovery. Institutions have been named in her honor. Awards celebrate her memory. Documentaries tell her story. But the recognition came decades late, after textbooks had been printed, after public understanding had been shaped, after generations learned about DNA without learning about her. The Nobel Prize ceremony in 1962 celebrated discovery. The speeches praised the model builders, and the woman who had seen the helix in her photographs, who had produced the evidence that made the model possible, was absent from the celebration, absent through death, but also absent through the structures of recognition that had never fully seen her. What does it mean to be forgotten? To contribute something essential and receive nothing in return? To see clearly and have others credit themselves with your seeing? Rosalind Franklin is now remembered. Her name appears in biographies and documentaries, in histories of science and studies of gender bias. Awards bear her name. Buildings commemorate her work. Students learn about her alongside the men who built on her contributions. The forgetting has been partly undone, but the forgetting was real. For decades, she was invisible, a footnote, an obstacle, a difficult woman whose data proved useful. The scientists who knew the truth, who understood that photograph 51 was her photograph, that the structural parameters were her measurements, that the evidence was hers, did not always speak clearly enough to correct the record. The institutions that should have protected her work allowed it to be shared without her knowledge or consent. The histories that should have credited her accurately told stories that minimized or erased her contribution. The pattern is not unique to Franklin. The history of science is filled with women whose contributions were attributed to men. men who were their supervisors, their collaborators, their husbands, their students. Lise Meitner, who explained nuclear fission, saw the Nobel Prize go to Otto Hahn alone. Jocelyn Bell Burnell, who discovered pulsars, saw her supervisors receive recognition. Chien-Shiung Wu, whose experiments disproved conservation of parity, was passed over for the Nobel that went to the theorists whose predictions she had tested. The pattern repeats across decades and disciplines, suggesting systematic bias rather than individual oversight. Understanding why the pattern persists requires understanding how recognition works in science, how credit is assigned, how histories are written, how some voices are amplified and others muted. It requires acknowledging that science, for all its claims to objectivity, is practiced by humans embedded in cultures with their own biases and power structures. It requires recognizing that correcting the record is not merely academic exercise, but necessary justice. Franklin's case has become emblematic. When people discuss how women's contributions to science have been overlooked, her name often appears first. Her photograph, that small image of an X-shaped pattern, that shadow of the helix, has become symbol of what careful work can achieve and what careless attribution can steal. But symbols can obscure as well as illuminate. Franklin was not merely victim. She was scientist of extraordinary ability who made important contributions to multiple fields. Her work on carbon structure, on DNA, on virus architecture, all represented genuine scientific achievements that stood on their own merits. She was not defined by what was taken from her. She was defined by what she accomplished. The recognition that comes too late is better than no recognition at all. But it is not complete justice. Justice would have meant acknowledgement at the time, credit given when credit was due, a place at the ceremony where her work was celebrated. What Franklin received instead was silence, followed by slow correction, erasure followed by recovery, a name written in water that generations have worked to carve in stone. Return now to the molecule, to the double helix that spirals through every cell of every living thing, that carries the instructions for building organisms from bacteria to blue whales, that connects you to your ancestors and will connect your descendants to you. DNA is not abstraction. It is physical substance, molecules that can be extracted and purified, that can be seen under electron microscopes, that can be sequenced and edited and transferred between organisms. The structure Franklin glimpsed in her photographs is the structure being studied and manipulated in laboratories around the world, the structure that underlies genetic medicine and biotechnology, the structure that makes possible both the promise and the peril of our genetic age. The promise is immense. Understanding DNA structure has enabled treatment of genetic diseases, development of vaccines, identification of individuals through forensic analysis, tracing of evolutionary relationships, modification of crops for improved nutrition and yield, The Human Genome Project, completed in 2003, determined the sequence of all 3 billion base pairs in human DNA, providing a reference that enables research into the genetic basis of health and disease. Gene therapy offers hope of correcting genetic defects. Personalized medicine promises treatments tailored to individual genetic profiles. The perils are also real. The same understanding that enables treatment of disease enables enhancement of traits, raising questions about genetic manipulation that societies have barely begun to address. The same technology that identifies criminals can identify anyone whose DNA is collected, raising questions about privacy and surveillance. The same modifications that improve crop yields can have unintended ecological consequences. The power to read and write the language of life is not power that comes with built-in wisdom about how to use it. Franklin did not live to see any of this. She died in 1958, decades before the Human Genome Project, before gene therapy, before CRISPR and the era of easy genetic editing. She saw only the beginning, the structure revealed, the implications recognized, the research directions opening. But the beginning was essential. Without understanding structure, nothing else was possible. The helix she photographed is the foundation on which all subsequent molecular biology has been built. The helix holds us all. It connects every living thing to every other living thing through the shared chemistry of inheritance, through the common code that specifies proteins across all domains of life. It connects us to ancestors who lived millions of years ago, whose DNA was copied and modified and passed down through countless generations to become our DNA. It connects us to descendants not yet born, who will inherit our genetic legacy and carry it forward into futures we cannot imagine. Franklin's contribution to understanding that connection, to seeing the structure that makes it possible, deserves remembrance, not because she was victim, but because she was scientist, not because credit was stolen, but because credit was earned, not because the story is sad, but because the achievement is remarkable. She saw what others could not see, and what she saw changed what we know about ourselves. The laboratories have grown quiet now. The X-ray tubes are cold. The photographic plates have been archived, preserved as historical artifacts in institutions that recognize their significance. Photograph 51 exists in collections, reproduced in textbooks and documentaries, its X-shaped pattern as familiar now as the helix it revealed. Rosalind Franklin has been dead for more than 60 years. The colleagues who knew her have also died. The generation that witnessed the DNA discovery is passing. What remains are records, notebooks and correspondence, publications and photographs, the evidence that historians examine to reconstruct what happened and what it meant. But something else remains as well. The knowledge she helped establish persists in every biology classroom, every hospital, every laboratory where the secrets of life continue to be decoded. The techniques she refined continue to be used, evolved and improved, but fundamentally similar to what she practiced in her basement laboratories. The precision she exemplified continues to be valued, the insistence on evidence, the refusal to claim more than data supported, the rigor that distinguishes science from speculation. She was not perfect. No one is perfect. She could be difficult, could be impatient with those she considered imprecise, could fail to navigate social situations that might have eased her professional path. The same directness that made her an excellent scientist sometimes made her a challenging colleague. The same standards she applied to her own work she applied to others, not always with sensitivity to how her judgments were received. But imperfection does not negate contribution. The qualities that made her difficult also made her capable of work that others could not do. The precision that could seem cold was also the precision that produced photograph 51, that revealed the helix, that provided evidence on which the model was built. She was who she was, and who she was produced results that changed human understanding. The light that lingers after her is the light of that understanding. Every time a doctor explains genetic testing to a patient, every time a researcher sequences a genome, every time a student learns about the double helix, Franklin's contribution continues. The recognition may have come late, but the knowledge she helped establish persists, accumulating, building, extending into applications she could not have imagined. Sleep comes now. The story closes, but it closes not with loss alone, not with injustice unremedied, but with something larger, with the spiral that connects all living things, with the structure that Franklin was among the first to see, with the knowledge that her careful eyes helped bring into being. She saw what others could not see. She captured in her photographs the architecture of inheritance. She contributed to understanding that has transformed medicine and agriculture and criminal justice and evolutionary biology. She was scientist of extraordinary ability, whose work endures even when her name is forgotten, and whose name is now remembered alongside the work. The X-rays have stopped. The photographs have faded, but the helix continues its silent turning, carrying forward the instructions for building everything that lives. And somewhere in that turning, in the elegant spiral of complementary bases, in the chemistry that makes inheritance possible, her contribution persists, unseen perhaps, but present, part of the foundation on which all subsequent understanding has been built. Rosalind Franklin, crystallographer, scientist, seer of structures invisible to ordinary eyes. May her memory be preserved with the precision she valued. May her story be told with the accuracy she practiced. May the helix that holds us all hold also her name, written not in water, but in the permanent record of what humanity has learned about itself. Rest now. The laboratories are dark, the photographs are archived, and the helix turns as it has turned for billions of years, as it will turn for as long as life persists on this planet, the hidden genius of DNA, forever part of what we know, forever deserving of remembrance. Good night. Conclusion. Where light breaks through. The story of Rosalind Franklin ends not with death, but with continuation. The continuation of knowledge she helped establish, the continuation of recognition slowly building, the continuation of the helix that turns in every living cell. She was not a tragic figure, though her life contained tragedy. She was not primarily a victim, though she was victimized. She was a scientist who did extraordinary work under difficult circumstances, who contributed fundamentally to one of the most important discoveries in the history of biology, who continued producing important research until illness made continuation impossible. The lessons of her story are multiple. That recognition is not always given to those who earn it. That the history of science reflects the biases of those who write it. That correcting the record requires vigilance across generations. That women's contributions have been systematically undervalued, and that conscious effort is required to ensure accurate attribution. But also that careful work endures, that precise observation reveals what speculation cannot, that the evidence eventually speaks, even when those who produced it are silenced. That truth, pursued with rigor and documented with care, survives the prejudices that tried to suppress it. Rosalind Franklin saw the shape of inheritance. She captured it in photographs that remain among the most important scientific images ever produced. She analyzed it with mathematics that confirmed what her eyes perceived. She contributed to understanding that has transformed human life in ways she could not have imagined. That contribution deserves remembrance. That story deserves telling. That name deserves speaking, alongside the other names that history has celebrated, with equal honor, with equal gratitude, with equal recognition of what was achieved. The double helix, the hidden genius, the light that lingers after. Sleep well, and wake knowing that the knowledge accumulated by centuries of scientists, including Rosalind Franklin, continues serving humanity, continues revealing the secrets of existence, continues building understanding that makes better futures possible. The story ends, the knowledge continues, and the helix turns forever, carrying forward the memory of all who contributed to understanding its elegant, essential form.